Hi everyone, this is lecture 6, reproduction at the cellular level. In other words, mitosis. So this comes from the Concepts of Biology OER textbook, chapter 6. So by the end of this section, this is hopefully, what you'll be able to do. Again, it might sound kind of like intimidating right now out of context, but hopefully by the end of this lecture, you will be able to describe what a genome is, describe the 3 stages of interphase, discuss the behavior of chromosomes during mitosis and how the cytoplasm divides during cytokinesis, explain how the 3 internal checkpoints occur at the end of different phases of interphase, and explain how cancer is caused by uncontrolled cell division. So right now this kind of makes no sense out of context, but hopefully by the end it will make sense and you'll be like, okay, yeah, I can describe what a genome is. Yeah, I can describe what the 3 stages of interphase are. So to begin, we begin life as a fertilized egg, or what's called a zygote is just the word for this fertilized egg. And so that one egg, when it's fertilized, it's one cell now. And so eventually that one cell has to divide to become a whole multicellular complex human. And so how does that happen? Through a lot of cell divisions. And so even when you go from being this one little cell to like a baby, and then a baby to an adult, you're doing this process of cell division just thousands and thousands and thousands of times to become like a full-grown adult. But even when a human is a fully grown adult, you still have to reproduce your cells and divide cells so that you can repair or regenerate tissues. So for example, we're constantly losing skin cells. We shed like 1,000 skin cells a day or something like that. And so we're not just like getting more and more like raw, gross meat, we regenerate those skin cells that we lose through cell division. So there are some kinds of cells that do divide pretty often to regenerate themselves, like skin cells or blood cells. So how do you do this? How do you go from one cell to multicellular and then make more cells? All multicellular organisms use cell division for growth, for maintenance and repair of cells and tissues. So yeah, a multicellular organism, like humans, to grow, we need to use cell division. And then even maintenance and repair of cells and tissues, we need cell division. And then single-celled organisms, so again, like bacteria, that just one cell is one organism, they use cell division as their method of reproduction. So they really just divide into 2, and then that's how they reproduce. So first, what is a genome? So all the DNA in a cell is called its genome. So we have a lot of different chromosomes that code for different things. And so when we think of DNA, we think of it as this 2-stranded molecule, right? Seen to the left on the bottom. So we picture the double-stranded DNA helix. And so if we were to take that and wrap it around proteins, that's what makes the chromosomes that we think of. We talk about, this, chromosome, or we have this many chromosomes. So chromosomes are all like your DNA, wrapped around proteins and then wrapped together to form this structure called a chromosome. So we actually have multiple pieces of DNA, and then each string is like really, really long and then organized into a chromosome. We have multiple chromosomes. And so all the DNA in a cell is called its genome. So you might have heard about the Human Genome Project. That's when we were trying to see, okay, what does all the DNA in humans code for? We have so many chromosomes. What do they all do? What does each gene on those chromosomes do? So that's what the Human Genome Project is trying to see what the code for was. for every gene on all those chromosomes. So that's your genome. If you were to take a cell and split and like dump out the nucleus and look at all the DNA in a cell is its genome. And usually it's organized into these structures called chromosomes. And so the number of chromosomes is different between species. So like depending on the species, you'll have less chromosomes or more chromosomes. So for example, fruit flies have 8 chromosomes because they're really small. They're really tiny. They don't have that much to code for. It would make sense you won't need like a whole bunch of genetic information for something so like little and simple. But humans have 46 chromosomes. We're very complex. We have a lot of different stuff to code for. So humans have 46 chromosomes in their cell. So even though the number of chromosomes is different between different species, within that species, all of its body cells are going to have that many chromosomes. So in fruit flies, all of their cells, they each have 8 chromosomes. And then in humans, each of our cells has those 46 chromosomes. And so here's a picture of what we call a karyotype to the right. And that kind of just shows laid out all the chromosomes that we have in our genome. And so again, this is another view of the karyotype and kind of how they make it. So they apply these dyes to cells and then they break open the nucleus. And so the dyes will kind of stain the different chromosomes different colors. So even though these two are matched, they are slightly different. So you get one chromosome of chromosome 1 from your mom and one chromosome of chromosome 1 from your dad, and that makes like one set of chromosome 1. And so they do this process and they align pairs, basically like one of those matching card games, based on the color that it gets dyed and then the length of the chromosome. They have to be the same length, and then that's a match. And then, yeah, that's how they can organize them into pairs. So these are a pair of chromosome 1, these are a pair of chromosome 2, and then 3, so on and so on. And so you can see the difference more between like chromosome 1 versus chromosome 22. Those are very different. So even though some of these lengths at the top look similar, they're all different, again, because they have different structures, they have different genes on them, and that's what makes them different chromosomes. And so I think of the genome and genes as like a cookbook. So each chromosome is like a special cookbook. Like this is the cookbook just for pastas, and then this is the cookbook for, you know, meat dishes or soups or whatever. So chromosomes have recipes for different things. And then you have 2 copies of each recipe. So that will get a little bit more into genetics. But yeah, your chromosomes are basically like the cookbooks or instruction manuals, people say, for everything that your cell needs to do and make and become. And we as humans happen to get 2, 1 from each parent. So that's why we have 2 of each of these different chromosomes, if that makes sense. And then we call each pair of chromosomes homologous chromosomes. So we'll revisit these concepts and this like idea in the next lecture. Right now it's just kind of laying out like, okay, genetic info is important and we want to make sure that each cell gets the right number when they divide. So the cell cycle is the way that your cells actually divide. So all cells come from other cells. Your cell and your body doesn't just put up like a scaffold and start building, you know, random pieces together to build a cell. All cells come from other cells. So one cell has to divide. It basically has to grow and double up on everything and then divides, and that's how it makes a new cell. So that is how, yeah, that's the cell cycle. That's how, again, things happen for like cell growth, for division, growth, and like repair of things, and asexual reproduction in organisms that do that. So they divide cells through the cell cycle. So you go from one cell, this first one, it kind of starts preparing, As we go through clockwise, it divides. So this is like a really shortened version of the cell cycle, but you start with one cell, divide, and then you end up with 2 cells. And then those cells will each end up doing the same thing. So to make more cells, each of those cells will end up dividing one day and then going through the whole process. So that's how your cells and your body makes new cells, through this whole process called the cell cycle. So the cell cycle has 2 major phases. And so each one, of course, like we can like simplify things in biology, but then there's also going to be more detail. But so to break things down first, the overall cell cycle is 2 phases. So interphase, so that's kind of shown by this outer circle in gray. So all of these steps outside is interphase. And then a small portion is the mitotic phase. So during interphase, this bigger portion of the whole overall cell cycle, the cell cycle is both of these things all together. So interphase is when the cell grows and kind of doubles up on everything and the DNA is copied and it's preparing to make all the stuff to have a second cell. And then mitotic phase or mitosis is when it actually divides. So yeah, we always think about like mitosis, but it is part of this bigger thing called the cell cycle. So interphase is these different steps where your cell is preparing and growing and copying DNA. And then the mitotic phase is when the copied DNA and the cytoplasm and all the stuff inside the cell is actually divided. So again, during interphase, to zoom in a little bit more, interphase is just this outer part shown in gray. And so interphase has 3 stages. So G1, G stands for growth. So the first growth phase is cell growth and then making proteins. So proteins, remember, one of the macromolecules, that's important for like a lot of different things. So basically the cell is growing and making all the stuff necessary to become 2 cells. That's G1. The S phase, S stands for synthesis. So the S phase is when you're making DNA. So you're copying the DNA that already exists. Each chromosome you're making like a copy of. And yeah, that phase is pretty important because you really want to make sure you get the DNA correct, because that is going to go into a new cell. If the DNA for that cell is incorrect that you copied, eventually when that cell divides, it's going to carry on that wrong DNA and just keep carrying on mutations. So DNA synthesis in S phase, when you're copying the DNA, it is pretty important. Your cell takes a while to do that to make sure it's doing it correctly. And then the last one is G2. Again, G for growth. So the second growth phase is just more growth and then protein synthesis. So yeah, your cell has to prepare and do all these things and copy the DNA and then make more proteins and things like that. So it's preparing. And if you notice how much more of the circle of the cell cycle interphase is, that's accurate. So your cells, when they're getting ready to divide, they spend a lot more time preparing to divide because they want to do it all right. They want to have all the correct materials before they do it. And then the division is actually pretty fast. It's just like a small portion of this overall cycle. So most of the time your cells are at some point in interphase in one of these phases. So either G1, S, or G2. And again, yeah, it's really important because you're kind of taking your time making everything that the cell needs to double up on everything and have the correct copies go to each cell. So your cells spend really like a lot of time in interphase. And then when those things are done, it goes to the mitotic phase, and that's actually pretty quick. So the mitotic phase, again, what is happening is that the DNA that you copied and all the stuff that you made actually gets separated, and then the cell divides. And then the mitotic phase, there's actually 2 things. So mitosis and then cytokinesis. So mitosis is when you separate the copied chromosomes that you made, and then you're dividing them up into the new cells. And then cytokinesis, we kind of think of happening for sure at the same time, but it is technically its own thing. So mitosis is kind of tracking the chromosomes and what would go into the nucleus of each cell. And then cytokinesis is the actual division of the cytoplasm and like the cell contents and stuff. So yeah, even though they're like lumped together in some of the following slides, mitosis is always followed by cytokinesis. So they kind of go hand in hand for sure, but they're technically separate. So hopefully I'll show that in the next slide. So again, this is like an overview. So the mitotic phase is made of mitosis and cytokinesis always. And then if we zoomed in on mitosis, there are different phases of mitosis. So here's a really nice picture that I like because it really simplifies things. Stuff in textbooks always gives like so much detail and it makes it overwhelming. So I like this picture by just simplifying it into like let's just track one thing and follow it through the steps, and then you can kind of add more detail later. So in general, yeah, whenever you're learning anything new, try to remember like the big picture. What's the point of mitosis? You're splitting one cell to become two cells. And then you could fit in the details. And so here, the first kind of level of detail is just tracking stuff. So if this is how we're just focusing on one cell and one pair of chromosomes. So again, say we're focusing on like chromosome 1. They're both blue. They code for the color blue, but like you get one chromosome from mom and one chromosome from dad. And so this is your starting material, and you want each of your cells to have the same material. So in interphase, remember that bigger chunk of the cell cycle, you copy the DNA. So here, the DNA is copied. Now you have two of the little light blue chromosomes and then two of the dark blue chromosomes. And to be able to divide up and be organized well, they pair up. So here they kind of look like X's. It's like two eyes that get like joined together at one point, and so they kind of look like X's here. So this is one of those chromosomes, this is the other of that chromosome because the DNA got copied. And then here the same for the blue one. So after interphase, the DNA is copied, the cell has already prepared all the proteins and stuff. So in mitosis, the first phase is prophase. I think it definitely helps to have mnemonics and things like that to remember each phase. So prophase, they pair up or they pre-pair. Prophase, the chromosomes pair up. So instead of being just like two blue chromosomes on their own, they pair up like this. And so when they pair up, we call those sister chromatids. So that's basically prophase. It's kind of just like, okay, those chromosomes are pairing up and it's preparing. Metaphase, the chromosome pairs meet across the middle. So again, that's how I remember metaphase, middle. So the chromosomes line up across the middle. And then there's these things, these kind of fibers that form, and it makes what's called the mitotic spindle. So I kind of think about it as like when Spider-Man's like shooting a web. So this is like the web being shot, and it grabs onto one half of the pair. And on the other side of the cell, there's a different, Spider-Man web, and they grab to the other half of the chromosomes. So metaphase, they meet across the middle, and the mitotic spindle forms. Anaphase, they get pulled apart. So these chromatids get pulled apart. And so it's like the web, he's like bringing it back. And so that's what's kind of like pulling the chromosomes away from each other, because they're each grabbed by a different web and then being like yanked opposite. So anaphase, they're pulled apart. And then telophase is the last step of mitosis. it's when the chromosomes are separated, and then cytokinesis is when the cytoplasm is getting separated. So mitosis is basically just following the sets of chromosomes, and then cytokinesis is the division of like the cell and the cytoplasm. So that's why it's kind of like a separate thing. So telophase and cytokinesis we kind of lump together, but it's telophase is tracking the chromosomes being separated, and then cytokinesis is when the actual cytoplasm starts like dividing off. So by the end, you have two identical daughter cells. So everything should get divided up equally, and each side gets exactly what they're supposed to get. So each of these daughter cells has a dark blue and a light blue chromosome, just like the original one did. And then the same for the opposite cell. So this one also has a dark blue and a light blue chromosome, and then now these cells, form their nucleus again and start becoming like a normal cell. So this slide I think is really helpful to show just kind of, again, the big picture and not so much small detail. So I think it's really helpful to maybe try drawing that out for yourself. Make like little circles. So here's prophase, here's metaphase, and then you could kind of divide them up for yourself. It's really helpful to just do it yourself so that you could see and kind of come to understand it better than just following a video. I think really like drawing it for yourself really helps. So this is kind of the simplified version, and then here's like a little bit more detailed from the textbook. So prophase is again when the cell's preparing and the chromatids pair up. So here's a little bit more detail with like the other stuff that's in the cell that obviously has to also get divided up. So normally DNA is it's loose. It is kind of just like a long loose string, and when your cell is getting ready to divide, then the chromosomes condense into the chromosomes. So it's like if you think about yarn, when you go to the store and you buy yarn, it's not just like a long loose mess. For easier transport and to get it home safely, it's bundled up into those yarn bundles. And so that's what chromosomes are. Usually our DNA isn't tightened up into that yarn bundle, it's kind of like loose. But during prophase, that's actually when the chromosomes condense into that yarn bundle, and then they can be like cleanly carried through. So that wasn't shown that much in the other one. It kind of just skips to that. But technically, yeah, your DNA is loose, and then prophase is when they condense into the yarn bundle because they're getting prepared to divide up. So here they get condensed into the little bundle called a chromosome. So chromosomes condense. Spindle fibers kind of start forming a little bit early, but in metaphase is when they like grab on. So the spindle fibers start emerging. The nucleus starts breaking down because you don't want a nucleus like in the way. You want to be able to kind of just cleanly divide up the the cell contents and the chromosomes. So again, that's why we organize them into chromosomes, because once you open the nucleus, they're going to be exposed to like all the other stuff in the cell. So chromosomes condense so that the DNA is protected through doing all this stuff. So the nucleus breaks down, and then that way the fibers can like grab on and then start pulling them apart. Prometaphase you can kind of skip. It's kind of just like, it's not really its own phase, but it's kind of just showing like some intermediate steps. So let's just skip to metaphase. I'm not going to have you guys memorize prometaphase. So metaphase, the chromosomes meet at the middle or across the middle. And then each half, so each sister chromatid is attached to a fiber, and then in anaphase they get pulled apart. Telophase, the chromosomes are at opposite poles. I haven't really come up with a good name for telophase, like a thing to remember it. Maybe like ta-ta, like they're already kind of separate, like that. So telophase They'll have to telephone each other because they're long distance now. But it's basically the last phase of mitosis. So the chromosomes are now at opposite ends, and they start decondensing, so they go back down to just kind of like the loose yarn because they are safer with the nucleus starting to develop again. And then the spindle fibers start breaking down. And then cytokinesis is when the actual cytoplasm divides and gets separate. And you can tell because there's, in animal cells, what's called a cleavage furrow, which is just, you can see like the little division. And then in plant cells, it's called something else, a cell plate. But either way, it's just terms for seeing there's a division in the cells being separate now. So those are the phases of mitosis: prophase, metaphase, anaphase, telophase. And yeah, again, I think it's helpful to just start and just focus on the chromosomes. So if you were to follow one chromosome pair and draw that out, and then you can apply all this other kind of detail. But if you just focus on the chromosomes, that's the most important part. That's what we care about specifically, you know, like the big picture. How do you divide a cell and make sure it gets all the right information? Because you have them meet across the middle and they get pulled apart so that each cell ends up with the right amount. So I really think like videos with animations really help because it's kind of boring and like static to just read about it. But when you have the animations and it shows how it happens, it's kind of helpful. Yeah. So if we go back to like zoom out to the whole cell cycle, so the cell cycle again, remember, has interphase, and then the mitotic phase, which has mitosis and cytokinesis. So again, it's super important to have everything copied correctly and made correctly, and then prepared to divide up correctly. So usually your cells have checkpoints at different parts of the cell cycle where it's really like a supervisor is coming in and saying like, okay, let's make sure that we actually have everything that we need, like, and then it kind of goes through a little checklist of things to make sure that that's correct. So the cell cycle has 3 checkpoints. So the G1 checkpoint, so right here, before you copy the DNA, after you've copied, you know, proteins and stuff like that, before you even copy the DNA, you want to make sure the DNA is fine. The integrity of the DNA is okay, because if the DNA is like messed up and there's huge errors, you want to know about that ahead of time before you make copies of it. So at the G1 checkpoint, it checks the integrity of the DNA. So like, okay, is the DNA fine? There's no like big mistakes. There's no huge chunks missing or like things that are wrong. Okay, if it's fine, then we can move forward. So then it can proceed through the S phase to make more DNA synthesis. And then after that, it goes through the G2 phase, some more cell growth, and that's another checkpoint. So at the G2 checkpoint, it stops and then again is like, okay, hold on. Let's make sure that we copied everything correctly now. So we made sure that the first copy, or the first like original, was fine. Let's double check that the copy that we made, no mistakes happened. You don't want like a game of telephone or something that like it gets like super messed up, because then that is just going to keep moving forward. So at the G2 checkpoint, they check that the proper chromosome duplication happened, there were no major errors, and if there are, then we got to fix them before we move forward. Either fix it, and if we can't fix it, then we got to just kill this cell. Yeah, so the G2 checkpoint is just making sure like, okay, proper chromosome duplication happened, everything's fine, it got copied correctly, okay, we can move forward. And then the last checkpoint that happens is during mitosis. The M checkpoint happens in metaphase. And so that makes sure that those spindle fibers, the kind of what I was saying, the Spider-Man web, that those actually attach correctly. Because say it didn't attach and it doesn't pull the right number of chromosomes into the cell it's supposed to, that's a big no-no. So at the M checkpoint, they make sure that the fibers grab onto each side of those sister chromatids. That way they can actually get pulled correctly to the right sides. So that's the M checkpoint. And so these checkpoints are really important. If your cells fail to pass through this, then it might be able to fix whatever it is and then proceed, or that cell will just get told, like, sorry buddy, you gotta go. We can't have this cell divide and lead to other messed up cells. Because that's what cancer is. So cancer is a collective name for a lot of different diseases. You know, there's very different cancer types. And they're all kind of started for different reasons. They might have different origins and like mechanisms. But cancer is the name of a lot of different diseases of these kinds because they all have a common mechanism, which is uncontrolled cell division. So there is a lot of, you know, like redundancy and these levels of cell cycle and 3 checkpoints, but even 3 checkpoints is not a lot. So some errors do occur, and sometimes they can be fixed, or sometimes they don't really amount to anything. But cancer is like a major mistake, and that gone unchecked becomes uncontrollable cell division. So like I said, one of the important steps of the cell cycle checkpoints is just making sure that proper copying of the DNA happened, proper replication of the DNA during the S phase. So that's a big deal because, yeah, if that DNA is mutated, especially depending on where the DNA gets messed up, if that happens to be in a gene that codes for something that takes part in the cell cycle checkpoints and regulation, so if there's a mutation in the gene that codes for something that's supposed to help keep the cell cycle in check, if that gets messed up, now the cell cycle can't be kept in check. And so it's kind of like a downhill, like, I don't know, snowball effect of, okay, now this thing that was supposed to limit and double check everything is not working. So now those cells are going to keep replicating and keeping on that mistake. Yeah, so if a change to the DNA sequence happens inside of a gene, this is called a gene mutation. And so we kind of want to prevent those. And the effects do depend on where a gene mutation happens, like in which gene it's affecting. So usually, all cancers begin when it's a gene mutation that makes a faulty protein that's usually part of the process of cell reproduction. So say there's like a protein that's in charge of being the supervisor and double checking, like, okay, how is the DNA looking before we even copy the DNA? Or how's the DNA looking now that we copied it? Like, is everything good? Say that a mutation happens to that protein that's supposed to be doing that. If all of a sudden it doesn't, then now this cycle is going on unchecked. And that's how this kind of keeps snowballing, basically. So even if the change is minor, even minor mistakes in those genes can allow future mistakes to occur more easily. So yeah, that is what makes the snowball effect happen. And then over and over, those small uncorrected errors are going to get passed on from the parent cell to those daughter cells and then just build up. So eventually, because of all these different like mutations and errors that build up, eventually the cell cycle speeds up because the control mechanisms decrease and the repair mechanisms decrease. So it's like there's nothing slowing down this cell cycle. So without all these other things keeping it in check, again, it's just snowballing. Like the cell cycle speeds up because there's nothing keeping it in check. So it's that uncontrolled cell growth of the mutated cells that ends up like outcompeting and outpacing the growth of normal cells in the area, and that is how a tumor can result. So that's why usually treatment options include like chemotherapy, because you're kind of trying to kill cells that grow really, really fast, like the ones that would happen here. And then, you know, you can have unintended consequences because you're kind of just targeting cells, and that might end up, you know, affecting your cells. But that is how uncontrolled cell division can lead to cancer, because if you mess with these checkpoints, it's going to just keep going and going and then happening faster because there's less and less keeping it in check. So that's why these checkpoints are really important. So the G1 checkpoint, okay, how is the original parent DNA? Is it good? Does it have any errors? No, it looks okay. Then we can move forward. And then the G2 checkpoint, how is the DNA that we copied? Does it look good? If not, let's fix it, or we just gotta like kill the cell so that uncontrolled cell growth doesn't happen. And then in mitosis, there is that third checkpoint where it's checking, okay, did we get the right chromosomes attached to the spindle fibers so that they get divided up easily. Yeah, so that is the cell cycle overall, the whole thing of interphase plus the mitotic phase, and the different phases of mitosis, and of course the importance of the different checkpoints. So again, yeah, all cells come from other cells. Like your body doesn't just put up a scaffold and make a cell from scratch. It starts with one parent cell, doubles up, and then divides into two. So you could think about that anytime you're like, anytime, especially if you like have a cut or something, you know, eventually your cut will like scab, and then end up, when it's done and your body's done making more cells, like that cut kind of goes away because your body made more cells by mitosis to replace those skin cells and, you know, complete your barrier again. And so if you ever look at like a baby, just think like, wow, it's going through mitosis right now. How does a baby go from like this big, you know, to like a full-size human? Through mitosis. Yeah. So now hopefully you can do all this stuff, you know, laid out in the learning objectives. So, you know, what's a genome? It's all the DNA in a cell. What are the 3 stages of interphase? G1, S, and G2, and what do they each do? Kind of discuss the behavior of chromosomes, how they get like divided up. Explain how the 3 checkpoints occur at the end of G1, G2, and during metaphase, and then how cancer is caused by uncontrolled cell division. Yeah, so hopefully now you feel confident in being able to do that, you know, with some studying maybe. But these are, you know, kind of your goals. So, yeah, see you on Canvas and in the next video for the next lecture.